Thank you, everyone, for coming here, not to my lecture, but to my home, which is the California Academy of Sciences, which also now hosts a nice exhibition of human evolution just behind you. So if you do not have the opportunity to see this new Human Odyssey exhibition, I encourage you to have a look at. I would like to thank the Leakey Foundation for sponsoring and for inviting us tonight to give a talk on diet, which I assume concerns every single person in the room today. Diet is today one of our, in spite of the calories, obviously, it's one of our key characteristics as a species that distinguishes us from many other species. I would also like to thank the staff of the California Academy of Science, including Gary, for doing such a fantastic job at every stage of this organization, of this series lecture. So thanks to the California Academy of Science and the Leakey Foundation, and obviously thank you for coming. I would also like to welcome my colleague, Christina Warinner, who will be talking more about what you eat, whereas I will be talking more about what you ate. Not yesterday. So, if you told me what you had was only 1 or 2 minutes, I would tell you the following things. 1, you can use carbon-stable isotopes to explore diet today and in the past in millions of years. The carbon isotope, the stable carbon isotope. Second, apes have C3-based, which is tree-based diet. Ardipithecus, Ramidus, and Animensis, Australopithecus Animensis, have more ape-ish diet, which means leaves and fruits. And then it's the species Australopithecus afarensis, which for the first time started to venture into more grass-based diet, described as C4 diet, which includes sedges and grasses, which also speaks to the type of environment they were frequenting in a way. And then by 2.5 million years ago, you see this dichotomy between what we call the Paranthropus, or the robust group, which became specialists of that hard, abrasive diet, or C4 diet, and then the genus Homo, which became more mixed feeder, probably because it involved with consuming a lot of meat with the invention of stone tools. But P. boisei, which is Paranthropus boisei, became the ultimate C4 consumer, including up to 80% of grasses and sedges or anything that may have been a C4 at the time. And then therefore, overall, you see the C4 diet being included as you go from the emergence of hominins, splitting from the chimpanzees sometime around 7 million years ago, all the way to today. And that was one of the key and fundamental changes that we observed as we explore human evolution. And that was happening concomitant with dental and jaw size and shape modifications. And all that was published only in June 25, 2013, on the cover page of the Proceedings of the National Academy of Sciences as 4 papers by many, many scientists, including myself. I don't see anyone moving, so I guess I will continue. But what are really the questions? As a paleoanthropologist, I like to think about, why do I do the job I do in the first place? Why is it important? And what are really the questions that we are interested to answer? Well, the first is we want to understand the anatomy of our body, not only today, but how it became what it is today. So anatomy and understanding how they looked, our ancestors, is important. That anatomy is going to affect or influence your mode of locomotion, the way you move around. And then combined, they are going to affect your behavior. And all of that would determine where you live, and where you live will determine your diet somehow, which then in turn determine your anatomy. So it's a circle that goes back into the origin of our anatomies. Therefore, diet is one of the key and fundamental issues in the history of human evolution, in the study of human evolution. If that is the case, therefore, what do apes eat today? And apes include many gorillas, chimps, humans, orangs, etc. What do they eat? Well, gorillas eat mostly leaves, and that is C3. I will come to the difference between C3 and C4 later. But they eat by and large leaves. Chimps consume fruits, even when they live in the savanna environment. They can live in the savanna environment, but they don't really consume grasses. They are by and large fruit eaters. Humans eat everything, and my colleague Christina will be talking a lot about that, telling you more. So my interest here tonight is, if gorillas do that, C3, chimps do, humans do everything, when did eating everything start? Is the question I'm asking tonight. But diet is really important because it's not only about the calories that we try to gain or lose. Homo sapiens are crazy. They try to do both ways. They take it in and they take it out again. They did not have that type of opportunity at the time. But it's not about the calories, but it is the interface between the organism, the humans or the human ancestors, and their environment. That's something that goes to your mouth. So it's going to affect, and it's going to be the source of a fierce competition between you, the organism, and your environment, and other species in that environment. Therefore, it's going to in turn affect the selective pressures that are acting upon you as you evolve through time. So it's beyond the calories. I mean, the issue of diet. So when did we start to consume grass-based diet, which is my talk today, or meat, or cooked food, etc., is really very important question in the study of human evolution. But how do we know then what an early human ancestor ate 3, 4, 5, 6 million years ago? There are many approaches that have been employed over the past few decades. The first being a very straightforward approach, that is to look at the gross anatomy of the fossils. You look at them if you have massive jaws and supporting muscles, like the nutcrackers, they were named, the Zinjanthropus boisei or Paranthropus boisei, then you would assume they were feeding on hard and abrasive diet. It's a pretty straightforward approach. Sometimes works, it sometimes may not. But by looking at the gross anatomy of a muscle, on the muscles, you can infer the type of diet consumed by an animal. Another approach is to look at the teeth size, shape, and topography. If you have sharp cusps like this, it would be used for shearing, say leaves. If you have more lower topography, then you would be mostly grinding maybe fruits like in pan. So this is another approach that also has been widely used to investigate diet in earlier hominins. Why? Because all we have in terms of fossils are the teeth and the bones. We don't have the soft tissues or the other evidence for the diet. You can also look into the archaeological record. If you have stone tools, then it must have served some purpose, maybe removing meat of the bones or butchering carcasses, etc. Or if you have cut-marked bones, it can show you that meat was being consumed by early human ancestors. So the archaeological record was also used to determine what an animal may have eaten 2, 3, 4, 5 million years ago. More recently, we've been investigating what we call dental microwear. These are those fine-grained structures that are left in your teeth by the food item you consume. So if you're feeding on hard and abrasive diet, you would have this very complex features with pits in them, whereas more scratches will indicate soft fruits and leaves. So these are techniques that have been used for many years to study fossil diet, but you can obviously see the limitations. So what we tried to do in this publication that we published a few months ago is to look into the chemistry of what the animals ate. This is a more direct evidence which would tell you the chemical composition of the diet that was consumed 3 million years ago by species A or C is going to be like this. Well, how do we do that? Well, first of all, we take samples up to 2 milligrams, and then we use a mass spectrometer to compare the heavy versus the light carbon isotope. How does it work? Well, as you know, there are two types of photosynthetic pathways. One is called C3, the second is C4, and C3 plants are basically the tropical trees, including that have leaves and fruits, bushes, forests, etc. The C4s are tropical grasses that have that also include sedges. And then you have the cam plants that are succulent plants and cactuses and pineapples that are mostly in mostly desert plants, but we're not concerned about this today. But how do we distinguish a C3, which is tropical trees, bushes, and forests diet, versus this one? I will come back to that. But please note that I'm not using radioactive carbon here. These are stable isotopes. It sometimes gets confused. Stable isotopes to investigate the food chemistry. Basically, what we're trying to do is, can we tell if an animal was eating, by and large, grasses and sedges, or tree-based diet, which would include fruits and leaves? That's the question. And let me take you, if you've forgotten your photosynthesis courses from high school or, I don't know, from college, this is how it works. I said C4 and C3, they don't have the same photosynthesis pathways. They have different pathways. The first one, C3, which includes most plants, and this has been evolving for millions and millions of years, whereas the C4 is a newcomer. What is C3? Photosynthesis is pretty simple. You have hydrogen, you have water and carbon dioxide. You use light to produce sugar. How do you do that? You fix carbon dioxide using an enzyme called Rubisco, which means ribulose biphosphate carboxylase. It's just an enzyme. And then it converts it into sugar using ATP and NADPH. These are basically the energy currencies of photosynthesis. So when you do that, you result in sugar, which has 3 carbons. It's called 3-carbon chain molecule. But what the problem with this Rubisco enzyme is, it's not only does it fix CO2, which is useful for producing sugar, it also fixes oxygen. And fixing this requires energy, so it wastes energy. It also has, and that wasting energy and then producing, fixing oxygen, which is not going to serve any purpose and sometimes is harmful, is called photorespiration. So that's why we call the C3 type of photosynthesis pathway is inefficient. Why? It's losing energy, and then it's fixing carbon dioxide, oxygen also. So the bottom line here is, because in the Calvin cycle, this enzyme is fixing oxygen, it's causing photorespiration, which means you're wasting energy by fixing oxygen, among other problems. Good. So to mitigate this problem, what the C4 plants did is, by the way, all that process, the Calvin cycle is happening here in the mesophyll, which is the upper part of the leaves. It's happening here. What these guys did is, okay, we're not going to use Rubisco, we're going to use a chemical called PEP, which means bisphenol pyruvate. And what it does is, it sucks the carbon dioxide, and then it produces a 4-carbon chain molecule called malate. And it's still not doing the Calvin cycle to produce sugar. Before doing this, it passes through this stage. And what is happening here is, it's getting the carbon dioxide from the air, it's producing a carbon molecule called malate, and then it sends the malate down to this special cells called bundle sheath cells, And then this carbon cycle process is happening deep in the leaves. What are the benefits? Well, first of all, the PEP does not fix oxygen. It fixes only CO2. So it's not wasting any energy. Second, because this is deep in the leaves, it's a closed system. Closed system means once you have the carbon dioxide inside from the mallet carries the carbon dioxide, there is no going out. So all the carbon that comes in is going to be used by the plant. Therefore, there is no carbon dioxide coming from outside entering here. So it's a completely closed system. As a result, you will have less heavy carbon 13 here, and you will have more heavy carbon 13 here. Why? Because there is a fractionation going on. There is a discrimination against the heavy carbon that's happening here, because if you have to choose light or heavy, you will choose a light because you can process it easily. It's like carrying something. But in here, because the system is closed, all the carbon that goes in is going to be processed. As a result, you will have more heavy carbon in the C4 plants as opposed to the C3 plants. This is the basics of the C3 versus the C4. So the crux of this point is C4 plants will have more heavy carbon. C3 plants have less heavy carbon. This is the basis of isotopic analysis. Given that, therefore, you can calculate the ratio between the heavy and the light carbon, which is the heavy is the 13, the light is this one, and then it's expressed in per mil. When you do that, C3 plants will have a ratio between minus 22 to 35 with an average of minus 2, minus 26, and C4 plants will have a value of minus 13 on average. As you can see, they don't overlap. Why am I talking about this? Because when I say C3, they are going to by and large be represented by grasses. So if I can identify in an early hominin that it has more C3, it was consuming more trees or fruits, leaves. If I have more C4, then it was involved in consuming grasses. That's why this process is very important. So these are the values, but there is further fractionation when you produce your enamel. So an enamel of a C3 feeding animal will be on average minus 13, so it will be more depleted. Why? Because to start with, there is less heavy carbon, whereas the enamel in the C4 feeding animal will have more heavy carbon. That's why it's a positive number. Actually, it could be negative depending on that. So again, to remind you, C3s are tropical trees, leaves, and fruits, bushes, forbs, and then here you have tropical grasses, sedges, and thanks to Christina, sugar canes also are included. That's why they produce very concentrated sugar. So that's the basics. So to recap, you have an atmospheric carbon dioxide, which is minus 7.5 per mil. Through the C3, you will have trees that are minus 26. As you go down up in the food chain, the browser will have an enamel with a minus 14. The predator will have exactly the same thing, and the same thing here. So giraffes who feed on leaves will have this type of value, minus 14, and wildebeest, which feeds on grasses, will have this type of values. Given that background, let me come back to what we did in our research. What we did is that we sampled hominin enamel cap from across Africa. And here are some of the pictures that you can see. And you really do minimal damage. If you can tell, this is before sampling, this is after sampling. And here is some of our team members sampling the 2 milligram or so enamel powder from this tooth. And that's all. The damage is really minimal. We did 11 species between 4.4 and 1.3 million years. These are all hominins. And then we've sampled 175 teeth so far. And the sample size has increased by 250%. This gives you an idea of what we did in terms of sampling. And I should underscore that what I'm presenting is the result of a collaborative work of all these people. And these are the titles of the 4 papers published in this journal a few months ago. So I am basically representing all these people in the list. You can see me here or somewhere around here. But this is a work conducted by many, many, many people. So I here stand in front of you representing all those people. So what are the results? So if we know that the C4 eating animals are eating grasses for the reasons I explained, the C3 eating plants are mainly based on trees, leaves, and fruits. What happens when we look? Oh, the results I showed you, by the way, are experimental on modern animals, so we know what those animals eat. Now let's look at the fossils from 3.5 million years ago, from the site of Hadar and Dikika, where I work at. So what are the giraffes doing? You can see the various giraffes, very negative values. It makes sense because they eat trees, leaves, and anyway, they feed on trees. Look at the wildebeest, very positive values. So this shows that the approach can be used for fossils first. These are the same age as Australopithecus afarensis. And what is afarensis doing? It's all over. It's consuming very C3-like diet all the way to C4-like diet. And there is a lot of variability. So it was a mixed feeder with considerable variability with an average of minus 7%, 7 per mil. This is really a neat result. Why is it a neat result? Well, because if you look at the Turkana data, the one I just showed you is from Hadar in Ethiopia. If you look at the data from the Turkana data, from the Turkana site in Kenya, look at Animensis, which is anatomically very similar to afarensis, is very depleted, i.e., it's feeding on C3 trees and leaves and fruits, like the chimpanzees, basically. But if you go further in time, here is time, from 4 million years all the way to 0, and here is the carbon content expressed as per mil. So Animensis is very ape-like. Pladiops, which is the same age as afarensis, is again very variable, but consumes both C4 and C3, so it's a mixed feeder. As you go up in time, Proanthropus aethiopicus, which is a robust megadont species, is continuing that C4 trend here, and Boisier takes it to the extreme, consuming up to 80% of grasses. Homo, on the other hand, which is us, 2.3 million years ago, continues to be a mixed feeder, which probably is due to the fact that they were including more meat. By that time, we have clear evidence of stone tool use. So if you are consuming meat of animals that consume trees, leaves, fruits, then you will have the same signature. If you put all the data together that exists today, you have Ardipithecus ramidus here, which is really chimpanzee-like. Very C3 diet. If you look at Animensis, very C3-like. Afarensis and Pladiops are the first creatures to really venture into the grassland environment, or at least exploit the resources they're in. And what's fascinating, I think, for me, is, so you have that, Africans continues that pattern, but what's fascinating is Boisier takes it again to the extreme of grass eating, whereas Homo does both. So why is this result important? Well, first of all, when we try to understand what makes us human, as I said at the beginning, it's anatomy, diet, locomotion, behavior, etc., environment, determining when that type of mixed feeding, which characterizes us, we take it to the extreme, obviously, emerged is important. Because the way to understand humans is to look at how we differ from chimpanzees, because we share a common ancestor 7 million years ago. So if we are to understand ourselves, when we split off from those species in terms of our locomotor adaptation when we became upright walkers, in terms of our dentation, which is tiny and small compared to others, in terms of our brain, which is huge compared to the chimpanzees. Those are the questions. But in terms of what we ate and when we started to venture into a different type of diet, this is the evidence for the first time that we're presenting in the study. Because it shows a major shift in terms of our adaptation when we look at the big picture of human evolution. So what was the source of C4 for A. afarensis? Well, clearly, they lived in woodland environment, and I'm sure there were grasses adjacent to that woodland environment. So they could definitely consume sedges and grass-based diet, et cetera, We cannot really pinpoint to that specific type of grass, but we have an idea that they were interacting with their environment differently, particularly compared to the chimps, who are always depending on the trees. When you always depend on a tree, when that tree disappears, then you disappear. I think afarensis ventured into this type of environment for the first time, which then ultimately continued to the genus Homo, then Heidelbergensis, et cetera, to create who we are. So this is really an important information as to what our ancestors were doing 3.5 million years ago. But in addition to the vegetation, if you remember, about 3 years ago, we published, my team from Dakika published, this cut-marked bones that showed that afarensis was the first creature to, well, in our history, obviously, the first hominin species to start using stone, to consume meat using stone tools. So it's possible, therefore, that consuming meat of C4 feeding carcass animals could also cause the increased amount of C4 content that you see in the species teeth. So a combination of meat eating and venturing into a new environment by about 3.5 million years ago is showing that afarensis was the first to venture into this type of environment. And when we published these results, we did not have the chemical composition of this species, but now it's coming together. So finally, the conclusions would be that Ardipithecus had very chimp-like diet. So Ardipithecus may have slightly shifted towards being human, but it kept small brain like the chimpanzees. It was still had grasping toe. It had a diet which was very ape-like, C3 diet, etc., etc. But aferens Ardipithecus, therefore, was, in terms of its diet, was chimp-like like Animensis. Animensis, in spite of its similarities in terms of its anatomy to afarensis, had not yet ventured into this type of diet either. So it was this species for the first time to really consume and go to the C4 world for the first time around 3.5 million years ago. And then meat consumption may have contributed to this fact. And then P. boisei, however, this is Paranthropus boisei, takes this adaptation passing through Ethiopicus to the extreme of consuming almost 80% grasses, at least C4 diet. And then Homo, therefore, continues to be a mixed feeder, including meat and more as we know it. And today, you know what happens. But there is a better person to speak about today, who is Christina Warinner. So I will stop here, and if you had questions, I will entertain them later. Thank you.